We said a minute ago that when we look at the composition of membranes, the most abundant lipid in the membrane is the phospholipid. But we said there are two other varieties of lipid that are also important to a membrane. The first are the sterols, and the second, the glycolipids. On this slide, you're looking at a depiction of an individual phospholipid here. You're looking at a little sliver of bilayer of membrane here. But this is the image I really want you to pay attention to. You can see we've got four individual phospholipids on the upper monolayer and four individual phospholipids on the lower monolayer. We're just looking at a little tiny sliver of a membrane here, a bilayer. The tails, of course, are all oriented towards each other. The heads are facing towards the outside of the cell and towards the inside of the cell, just like you would expect. What I want you to notice are these little yellow molecules here, what are tucked in among the tails. These are depictions of a sterol, and they happen to be representative of cholesterol. Cholesterol is the most abundant sterol that we find in the membranes of animal cells. And the most sort of glaring structural feature of sterols is the four-ring structure that you see here. 1, 2, 3, 4 rings joined together, making up this sterol. Now, the other type of lipid that we find in membranes is the glycolipid. And I said a minute ago that you can think about glycolipids as being phospholipids that are modified with carbohydrate, short chains of carbohydrate that are attached in the head region of the phospholipid. Now, structurally, it's a little more complex than that. So let's talk about the specifics of the structure of a glycolipid. So remember, we would normally expect a phospholipid to have glycerol and phosphate in the head and two fatty acids as tails. In a glycolipid, the carbohydrate that gets added is actually going to substitute for the phosphate group. So we're going to remove the phosphate group and substitute carbohydrate instead. The glycerol that we would expect to see in the head is going to be substituted with that molecule called sphingosine. And because sphingosine has essentially a fatty acid within it, we're only going to need one additional fatty acid. So this molecule, this glycolipid, is a modified phospholipid. It doesn't have a phosphate group. It has carbohydrate instead. It doesn't have a glycerol. It has sphingosine instead. And it has one additional fatty acid, so that you end up with two tails, but one tail comes from sphingosine and one tail is its own fatty acid. On this slide, you can see a depiction of one of these glycolipids. The way we typically draw glycolipids is, instead of drawing a circle for the head and two lines below representing the fatty acids, we draw a hexagon for the head and two lines below representing the fatty acids. The hexagon reminds us that up in the head region there's carbohydrate now instead of glycerol and phosphate. And as you can see in this drawing, the carbohydrate that you find in glycolipid really varies. There are several different varieties of glycolipid. So everything from a simple sugar being added on as the carbohydrate to a disaccharide here, to what you might call an oligosaccharide here with several sugars. And this one, which has 1, 2, 3, 4, 5, maybe 10 or 12 individual sugar molecules attached, which you could get into the polysaccharide area here. These are all depictions of different varieties of glycolipid with differing amounts of carbohydrate attached. This drawing in the middle is showing us that this is not a phospholipid. This is a glycolipid. It doesn't have a phosphate group. It has carbohydrate. It doesn't have a glycerol. It has sphingosine instead. And this region right here belongs to the sphingosine. It looks like a fatty acid. It functions like a fatty acid, but it's actually a portion of the sphingosine molecule. And here's the second fatty acid that gets attached. So glycolipids, for structurally speaking, act like phospholipids. They look like phospholipids for the most part. But internally, the details of the glycolipid are different from a phospholipid. Remember, we said there are two varieties of glycolipid that we're going to be talking about. One are the cerebrosides, and the other are the gangliosides. Now, cerebrosides and gangliosides are mostly found in nervous tissue. So we're talking about cells that are found in the brain, cells that are found in the spinal cord, cells that are out in the peripheral nervous system. They tend to be high in glycolipid. Now, the only difference between a cerebroside and a ganglioside is charge. It turns out that in order for a glycolipid to be considered a cerebroside, it has to be a neutral molecule. And in comparison, gangliosides have a net negative charge associated with them. That's the only difference between those two types of glycolipid. Glycolipids are critically important to the normal function of membrane inside the nervous system. That's why they are present there. That's why they are abundant in nervous tissue. But like all parts of a living cell, glycolipids have a limited lifespan. And like all parts of a living cell, glycolipids will be recycled. They will be degraded and recycled when their functional use is done, is over. Remember, we said there's an organelle in living eukaryotic cells that does the job of degrading molecules that are finished in terms of their functional life. And that organelle is the lysosome. Lysosomes take in these biological molecules within the cell when they are no longer useful to the cell, when their lifespan is through, and the lysosome breaks them down using that particular type of enzyme we mentioned called the acid hydrolase. The glycolipids will be broken down so that their pieces and parts can be recycled in the cell and used to build new molecules. And in a normal, healthy, functioning human being, that happens every single day. Glycolipids get sent to the lysosome breaks them down, degrades them, and recycles the pieces within the glycolipid. But there is a disease that happens when cells are not able to do this normally. It's a very interesting disease, and it's a good reminder for us about how important it is for cells to have functioning organelles. This disease is called Tay-Sachs disease, and you may have heard of it before. This is sort of a very famous, although rare, hereditary disease. And it's a disease of the lysosome. It's called one of the lysosomal storage diseases because there are other diseases that affect the lysosome's functioning, but Tay-Sachs is probably the best known. Now, Tay-Sachs is a rare disease, thank goodness. It tends to be found in certain populations around the world that have concentrated, unfortunately, this particular genetic defect. For example, there are groups of people of Eastern European descent, particularly a group of people called the Ashkenazi Jews. And this particular population of Jewish people tends to carry this genetic defect. There are certain French Canadian communities that carry this defect. There is at least one particular Amish community that carries this genetic defect. There's a community in Louisiana of people of Cajun descent who carry this defect. It's a terrible, terrible disorder. It affects children, and it is lethal because you cannot live without functioning lysosomes. So let's talk about what happens when a human infant is born with Tay-Sachs disease. These infants begin to develop perfectly normally, and during the first year of their life, they tend to hit all of their developmental milestones. They, babble with their parents, and they smile, and they eat normally, and they sleep, and they eventually learn how to sit up and to crawl and then walk. And then things begin to change. What's happening inside the nervous system of these children who have inherited this disorder is that their lysosomes are filling up with glycolipids. Glycolipids that are sent to the lysosome at the end of their normal lifespan. So as nervous tissue needs to be replaced because it's come to the end of its lifespan, and the lysosome encounters these glycolipids, it cannot break them down. It needs an enzyme, and this particular enzyme that's involved is called hexosaminidase. I sometimes stumble over that pronunciation. Hexosaminidase. And this enzyme is critical for just breaking apart this molecule called a glycolipid. What happens when the lysosome can't break down the glycolipid is that the glycolipid just builds up inside the lysosome. It just sits there. And over time, again, month after month after month after month, as this membrane arrives at the lysosome and the glycolipids pile up, the lysosomes eventually become packed with glycolipid. And once they're full up of glycolipid, they can't do their normal job. Any other materials that arrive at the lysosome for digestion have nowhere to go. There's no room in the lysosome because of the buildup of glycolipid. And so all of this material that needs to be degraded inside of these cells can't be degraded. And you start to see cell death. And these children in their second and third and fourth year of life start to regress. Again, particularly related to the nervous system. So you start to see a loss of motor skills. You start to see children who were crawling, who were sitting up and starting to walk, regress. They start to have symptoms of inability to integrate certain sensory input. They startle when they hear loud sounds. They start to lose their sight. They start to lose their hearing in general. That progresses on to seizures, muscle weakness, and eventually, unfortunately, death at a very early age. It's all related to a genetic defect in this enzyme in the lysosomes and an inability to break down a molecule called a glycolipid. Beyond the phospholipids and the glycolipids, remember the third type of lipid that is abundant in membrane is the sterol. I mentioned cholesterol a minute ago. Cholesterol is the main sterol found in animal membranes. Sterols in general have a structural feature in common. So it doesn't matter what kind of sterol you're talking about, it has this very characteristic structure. These 4 rings. You can see that 1, 2, 3 of the rings are 6 carbon rings, and the last one is a 5 carbon ring. Remember, when we draw a ring structure like this, each point of the ring represents a carbon atom. All right, so this is a 6 carbon ring. We haven't drawn in the carbons, we haven't drawn in the hydrogens that are attached around the ring, but the ring itself represents those atoms. So you have this 4-ring bulky structure and this hydroxyl group attached. This is what all sterols have in common. Now, cholesterol sort of gets a bad reputation because dietarily, a lot of human beings tend to overeat cholesterol, and it can cause problems in the cardiovascular system. But it's important to know that cholesterol is an absolutely essential molecule for the normal functioning of membranes in animal cells. It helps to maintain and stabilize membranes. The other thing that sterol is critical for, particularly this molecule called cholesterol, is the synthesis of steroid hormones. And a great example of steroid hormones are the reproductive hormones. So estrogen and progesterone and testosterone, these are the reproductive hormones, and they are all built from cholesterol. So cholesterol is a critical molecule inside normal, healthy, functioning animal cells. Plants don't have cholesterol, which is part of the reason that, again, when human beings get in trouble consuming too much cholesterol, they're often told to limit the amount of animal products that they're eating and instead start eating more plant-based products. That's because there is no cholesterol in plant membranes. Instead, there's a sterol called phytosterol. Now, I did put a note on the slide. There are a couple species of plant that have a little bit of cholesterol in their membranes, but don't worry about that. The most abundant sterol in plant membranes is phytosterol. The fungi have their own variety of sterol as well, called ergosterol. So it doesn't matter if you're talking about animals or plants or fungi, they all have their own version of a sterol that helps maintain and stabilize their membranes. I have a note at the bottom of the slide about bacteria. Bacteria don't put sterol into their membranes. They put a molecule that looks a lot like a sterol in, but you should know that the domain of bacteria, generally speaking, don't have sterol in their membranes. Another interesting difference between the domains. On this slide, up at the top, you're looking at that basic structure again of a sterol. We have 4 carbon rings attached to each other and a single hydroxy group coming off of one of those rings. So all sterols have that basic structure. Now, if you look at this drawing here on the left, this is the structure of cholesterol. You can see the 4 rings here. Here's the hydroxyl group. And then you see all the modifications that make cholesterol a unique molecule. If you compare that to phytosterol, you can see the 2 molecules are different from each other based upon these additional groups that are added on. You can still see the 4 rings of a sterol in the hydroxy group, but the additional functional groups that are added on make phytosterol different. And here's ergosterol down at the bottom with its unique chemistry. Now, a couple of other details to know about sterols. When you look at the organelles in eukaryotic cells that have a double membrane, and remember we said that is the mitochondria and the chloroplasts, the inner membrane of those organelles is largely free of sterol. Now that makes sense in light of endosymbiotic theory, right? Recall that endosymbiotic theory tells us that these double membraned organelles arose through evolution when a larger primitive cell consumed a smaller primitive cell. The smaller primitive cell then had a plasma membrane around it that then became the inner membrane of the mitochondria and the inner or thylakoid membrane of the chloroplast. Because that life form, that small primitive cell, was what it was a primitive organism, it didn't have sterol in its membrane. That would have been the type of organism that either predated the bacteria or perhaps was part of the early bacterial domain, and it would not have had sterol in its plasma membrane. So once it became engulfed and became the inner membrane of the mitochondria, the thylakoid membrane of the chloroplast, it didn't have sterol inside of it. And that is still true today in modern time. Now, I mentioned to you that bacteria don't have sterol in their membrane, but they do have a similar molecule. And you should know the name of that particular molecule. It's called a hopanoid. We believe that hopanoids are simply a more primitive form of sterol. So the hopanoids would have evolved first, and they would have been part of bacterial membrane and served that same function in bacterial membrane to maintain and stabilize those membranes. You're looking at a hopanoid over here on the right hand side. Again, you can see it's different from a sterol. We have 5 carbon rings instead of 4. There's no hydroxyl group off to the side. We have some methyl groups instead. Again, in all likelihood, the hopanoid came first, and eventually the sterol evolved out of the hopanoid. I'd like to switch gears here and start talking a little bit more about the function of the membrane, particularly permeability. So let's start just with a review of some of the general characteristics that you probably already know about membrane permeability. Let's start with a definition of this term. When we talk about permeability, we're talking about a measure of the ability to let substances pass through. So membranes are often described as being less permeable which would mean they have less ability to allow things to pass through, or more permeable, meaning that things pass through them with greater ease. Membrane permeability is going to vary from cell type to cell type, from organism type to organism type, from domain to domain. Membrane permeability is greatly affected by the structure of the membrane, by the types of phospholipids that are in the membrane, the amount of glycolipid, the amount of sterol. Membrane permeability is also impacted by a critical environmental measure, which is temperature. So let's start with temperature. When we're thinking about temperature, remember, we're really thinking about kinetic energy. We're thinking about how much individual molecules are vibrating. At lower temperatures, we see lower kinetic energy in the membrane, and the molecules within the membrane are vibrating at a slower rate. At a higher temperature, molecules are vibrating much more. So, at a low temperature, relatively speaking, when there's less kinetic energy in the membrane, when molecules are vibrating less, what that means is we're going to see a decrease in the ability to allow substances to pass through. Decreased permeability, in other words. Everything is moving less, so it's less likely that a substance could pass across the membrane in that state. Now, the opposite is true when there is a high environmental temperature around that membrane. When there's a high environmental temperature, the kinetic energy is higher. Molecules are vibrating more in the membrane. They're moving more. Think of that as the membrane having a more fluid state, and it becomes easier for things to pass through. In other words, the permeability increases. So temperature, depending on whether you're talking about a low temperature or a high temperature, is going to impact how easily things can get across the membrane. Now, there are three structural features of a membrane that will impact permeability, and all three of them relate to the tail region. The first is the amount of cholesterol in the tail region. The second is the amount of saturation in the tails. And the third is the length of the fatty acids in the tails. Let's start with cholesterol. Remember, cholesterol is a big, bulky molecule. So when you start adding cholesterol to the tail region of a membrane, the cholesterol is a space-occupying molecule. It is literally just taking up space that exists in between the tails off of the phospholipid molecules. So the more cholesterol there is in a membrane, the stiffer that tail region is, just because there's less space. So think of a membrane that's loaded with cholesterol as being less fluid. It's stiffer, it moves less, it takes up space, and so it becomes harder for a molecule to go across that membrane because there's just no space for it to travel through. It's going to bump into cholesterol. So the higher the amount of cholesterol, the lower the permeability. And the opposite is also true. The lower the amount of cholesterol in a membrane, the higher the permeability. Now, I'm talking about cholesterol here, but if we were talking about plant membranes, you could substitute in phytosterols If you were talking about a fungal organism, its membranes you would substitute in ergosterol. We're talking about the sterols in membranes here. Now, when we talk about saturation of the tail region, we're talking about the presence or the absence of those double bonds between carbon atoms in the hydrocarbon portion of the tail. Remember, each one of those phospholipids has two tails, and the tails are fatty acids. The fatty acid is made out of a hydrocarbon and a carboxyl group at one end. So within the hydrocarbon, which is just a long chain of carbon atoms bound to hydrogen atoms, if you add double bonds anywhere in that long chain, the molecule is going to bend, it's going to kink, and that kink introduces space in the tail region. When there is double bonds in a tail, we say that that tail is unsaturated. When there are no double bonds, we say that that tail is saturated. So, depending on the saturation in the tail region of a membrane, you'll see changes in permeability. The increased number of double bonds, meaning less saturation, will increase permeability in the membrane. Once you add double bonds, you're talking about unsaturated tails, you will increase permeability. And that's because you've introduced space, physical space, in the tail region because of the kinks in the tails. Now, the opposite is also true. If you take away double bonds in a membrane, you are saturating those tails. And if you have a higher amount of saturation, you're going to have a lower permeability. Higher saturation of the tails means less space. And the less space there is, the harder it is for something to come across that membrane. Now, the last structural feature in the tail region of a membrane that impacts permeability is the length of the fatty acid tails. It's possible to increase the length of the hydrocarbon portion of a fatty acid in a membrane, and when that happens, you're going to decrease the permeability of the membrane. So the longer the tail, in other words, the longer the fatty acid is, the lower the permeability is. Now let's think about why that happens. Remember that the tail regions of a membrane are hydrophobic, water-fearing, non-polar. And what holds the tails together are the weak attractions that we call van der Waals interactions. The longer those tails are, the more van der Waals interactions there are. In other words, the more tightly the tail region is sort of bound together. And the more van der Waals interactions there are in the tail region, the harder it is for something to come across. The lower the permeability, in other words. Now, the opposite is also true. If you shorten up the tails in a membrane, if you make the fatty acids shorter, that means there are just going to be fewer van der Waals interactions holding those tails together. And the fewer the van der Waals interactions, the higher the permeability. The easier it is for things to come across that membrane. So cholesterol or sterol amount, degree of saturation, and length of the fatty acids, all three of those structural features of the tail region will impact the permeability of a membrane.